Hello everyone. We are here with our next topic, intermediate filaments. It is a type of cytoskeletal elements that is usually found in vertebrates and lower metazoans, including mollusks, nematodes, etc. But they are absent in case of plants and animals. As I have discussed earlier, that these intermediate filaments, they have diameter in between microtubules and microfilaments, that is they have average diameter around 10 nanometer. They are basically non-polar and are made up of number of different types of proteins, and these are heterogeneous proteins, and they can be keratins, they can be type of lamins, they can be type of neurofilaments. But as a whole, as I have told you earlier, that cytoskeletal elements, microtubules and microfilaments, they have important roles in cell motility. But here, intermediate filaments, they have no role in cellular motility. But one thing which is common among these different heterogeneous protein filaments is that they share a common type of structural organization. But these, as a whole, these intermediate filaments, uh, their proteins are chemically heterogeneous, but they show species dependent variations with respect to their molecular weight. But the principal functions of intermediate filaments are structural, to reinforce cells and to organize cells into tissues. Unlike microtubules and microfilaments, they do not participate, as I have told you earlier, that they do not participate in cell motility, but they have individual polypeptides which share common type of structural organization. The individual molecule of intermediate filament is a is an elongated molecule consisting of non-alpha helical and terminal. It is non-alpha helical and terminal head domain and a central. This is a central alpha helical domain. This is central alpha helical domain, and it is central one, and it is having a non alpha helical. This is also non alpha helical C terminal domain. So the individual polypeptide of intermediate filament, it is having non-alpha helical head domain, alpha helical central domain, and non-alpha helical C terminal domain. This is C terminal domain, and this actually form a monomer of intermediate filaments, and here they first parallelly coil around each other to form a dimer, where non-alpha helical head domains are placed at the same end of dimer, and C terminal non-alpha helical domain are placed towards C terminal side or the same side of a dimer. Then here comes a phenomena which is different from microtubules and microfilaments. Here these dimers, they are further arranged to form tetramers, but the arrangement of dimer to form tetramer, it is anti-parallel. Means here there occur mixing of N terminal domain of one dimer to the C terminal domain of other dimer. This is blue colored dimer. Here it is having its N terminal ends, and here it is red color dimer expose its C terminal ends towards N terminal end of first. So here polarity should be like this, but here this is polarity, means N terminal N terminal on the same side, C terminal on the same side. But here what happened, the N terminal and C terminal, N terminal is here and C terminal is here, N terminal is here and C terminal here, and it becomes non-polar. And due to these tetrameric structures having mixed arrangement of N and C terminal ends of dimers, it forms non-polar anti-parallel tetramer. And these non-polar tetramers, they are arranged to form intermediate filaments, which is a type of polymer from of these tetramers. And these intermediate filaments, they are then having diverse roles in different cell types, as they are explained in subsequent points of the topic. But here important task is they loss there is loss of polarity with the formation of these tetramers. Up to monomer and dimers, they were polar, but as soon as they are anti-parallelly arranged to form tetramer, the polarity is lost. Now these tetramers, non-polar tetramers, they are arranged one above the other to form a polymer that is called as intermediate filament. So this is the structure of intermediate filament. Here C terminal end and N terminal end, they are fused on, and this is other end. N terminal end of one protein and C terminal end, they are found on both, they are found in mixed position, in mixed form at both ends of the filament. So it means these intermediate filaments, they are non-polar. There are four different types of intermediate filament on the basis of type of proteins, and in the type of tissues these intermediate filaments are distributed. They are nuclear intermediate filaments, they are bimentine like intermediate filaments, they are epithelial intermediate filaments, they can be axonal intermediate filaments. First type, it is nuclear intermediate filaments. They are having proteins nuclear lamines. Learn, it is lamine, Lamins, lamines. They are type of A, B and C. Basically these forms nuclear, they are found in nucleus beneath nuclear envelope. Nuclear envelope. They have role in forming structural network. They have role in forming structural network within nucleus, and it give a definite shape to the nucleus within the cell. And second type of intermediate filament is bimentine like. They are most diverse type of intermediate filaments, as they are found both in case of eukaryotes as well as in prokaryotes. In case of bacteria, these bimentines, they have been observed. On the other hand, in animals, they play important role in leukocytes, mesenchymal cells, and they are also leukocytes, and they are also expressed in endothelial cells of blood vessels. They have diverse different roles in these different types of cells, and basically they are non-contractile in nature. Non-contractile in nature, and they helps in aggregation of leukocytes. And third type of intermediate filament is epithelial. They are type of That means there are different types of keratins, keratin type 1, type 2, etc. And they are expressed in a number of cells such as skeletal cells, cardiac cells, and smooth muscles. And these are basically Desmond's types. And keratins, on the other hand, type 1 and type 2, they are obligatory heterodimers which helps in formation of features, structural features in animals. And fourth type of intermediate filaments is axonal filaments, and they are rich in proteins, neurofilaments L, NF, that is neurofilament L, neurofilament M, neurofilament H, and they basically determines shape of cells and maintains axonal transport. That is their main important role. And collectively, these are unbranched. They form polymers which are unbranched. So this is all about intermediate filaments, their structure and types, and in which type of cells they are found. And in subsequent lectures we will discuss about their regulatory functions in animal systems. If you like my video, you can share it, comment on it, and can subscribe my YouTube channel for many more such videos. Thank you.